So the second use case that I have is if you can't find any literature on a particular variant, how do you find information that may be helpful for variant interpretation? So I'm back on my home page. I will perform another search, this time for the ATM gene, a particular variant, the D1853P variant. If I perform that search, you'll notice that this results page has a different configuration. Over here on the right, where before I had article results, now I'm displaying information that suggests that Mastermind, despite its exhaustive and comprehensive search of the literature, was not able to find that particular variant, no matter how an author could describe it, in its indexing. And it allows you the ability to create an alert for that search. If I click on this button, it'll take me to the alert page, and I'll be allowed to receive emails weekly, monthly, or quarterly with any new information that's published about that particular variant. So that's one capability that you have if your results return no articles that mention that variant. On the left-hand side of Mastermind, it allows me to showcase the exhaustive search that we performed of the ATM gene, just as we've done for all other genes. That is the variant landscape for the ATM gene. And you see each of those peaks on the Manhattan plot is an individual variant. The x-axis depicts the linear axis of the protein with functional domains in the protein, the fat domain and the PI3 kinase domain in ATM, so that you can get a sense for what functions of the protein the variant may be influencing. And then the y-axis for each of these variants is a reflection on a log scale of the number of articles that mention each of those variants. So if I hover over, I can see the entire landscape of those variants. If I'd like to zoom in on any of that content with 2 fingers on my touchpad, or I think a right click and a scroll down on a PC, you can see that content expanded out, and then I can reset that capability to bring that back all on screen. So that's a visual depiction of the landscape of variants, which is very useful if you're, say, recognizing a loss of function mechanism for ATM, or in the case of a BRAF gene, which I'll search on later, you can see hotspot regions that are recurrently mutated. Down below, just as we had for the article view, you had a visual representation and a listed depiction of that content, are all of the variants in the ATM gene that Mastermind has found in accordance with the number of articles that mention that particular variant. So in this case, our variant returned no search results, but our interest is in finding related variants. So one thing that we can do is filter that list by the position. So I've typed in 1853, and now what I'm able to see here are variants that aren't the same variant as what I had searched for originally, but that are related by virtue of the residue being mutated. And you can see all of the different numbers of articles that cite each of those variants, and you can click on each one iteratively to see which of those variants is pathogenic and may then influence the likely pathogenicity of this D1853P variant, for which there was no evidence in the literature. While we're on this feature, there's other ways that you can organize this data to address specific questions about the variant, about the gene or its variants. I can say identify all the frameshift mutations in this gene by typing in FS. I can see all of the splice acceptor variants by typing in SA, the splice donor variants by typing in SD, all of the deeper intronic variants by typing in INT, as well as looking at, say, DEL or INS variants. One other capability that you have here when you've performed a search is by sorting after filtering that list of variants by cDNA position, so that you can see, say, all of the variants that appear in the 5' region that's removed from your particular search, or otherwise variants in the neighboring vicinity of your particular variant that may not be right at that particular residue. So that's a useful capability, either eyeballing it and getting a sense for the landscape of variance in the variant diagram, or otherwise using more explicit filtering and sorting capability in the variant pane.